Welcome to Biowise with Aanchal. Today we are going to study about Mendelian cross, laws of Mendel, and the monohybrid and dihybrid cross. It is on the request of a student who has mentioned in the comment section that he wants an explanation on this topic. So before proceeding, like, share, and subscribe the channel so that it can reach to the maximum who needs it. Now, before proceeding with this topic, you should be aware that what kind of question is asked in the examination. Now, on the basis of this question, MCQs can be many. Now, the most basic question that is asked is upon the ratio. Now, without doing the crosses, you should know that if you're talking about a monohybrid cross, then in F2 generation, the ratio is 3:1. And that in a dihybrid cross, the ratio is 9:3:3:1. How? We'll see it in just some time. Okay. So I'll be explaining you the sort of question that is asked and how you can be quick in writing the answers. So let us proceed with the unit. Here you can see that we are talking about the monohybrid cross. Before knowing about the cross, you should know that in monohybrid cross, we talk about one character. And in dihybrid cross, we talk about multiple characters. Okay. So here, the 7 characters that we are talking about is the 7 dominant traits. How will you remember the 7 dominant traits? You remember the mnemonics go in party. Now here go in party, go and in. Okay, go and in are for the pods. We are talking about the pods here. But party is about the plant, flower, and the seed. Okay, so green inflated pod is a dominant trait. Purple flower, axial flower. Then you have round seed, tall plant, and yellow seed is party. So go in is for the pod and party is for the other traits. So there are total 7 traits that we are going to, 7 characters that we are going to study. What is the difference between a character and a trait? Now, when Mendel was studying about the pea plant, that time genes word was not discovered or nobody has coined the term that time. 1909 mein Johansen has coined the term genes, right? But his work was in 1856. It's long time back, right? That time he used the term character, factors, and traits. Okay, so character is equivalent to the gene and the traits is equivalent to the alleles, if you can understand that way, right? So he said that the 7 characters he has studied about the plant, that is flower color, seed shape, seed color, pod shape, plant height, and flower position. So these are characters, and they all have 2 contrasting traits. Like flower color is purple or white, seed color is round or wrinkled, seed that is seed shape. Seed color is yellow or green. Pod color is green and yellow. So here is a point of confusion. So go in is for the pod and green pod is a dominant trait. Pod shape is inflated and constricted. Plant height is tall and short, and flower color is flower position is axial or terminal. Axial means at the axillary bud in between the branches, and terminal is on the top of the plant. So that is what the position of flower is all about. So let us move ahead with the law of dominance. Now, law of dominance states that out of the contrasting traits, out of the contrasting traits of a character, here only one character is studied, right? So present together, only one is able to express itself, while the other remains suppressed, and the one which expresses itself is a dominant trait, and the one which gets suppressed is a recessive trait. So dominant trait expresses in both homozygous and heterozygous condition, whereas recessive trait can express only in homozygous condition. Okay, so in this example, you can see that for a pea plant, tall is a dominant trait over dwarf, that is short plant. So for a genotype, if you say that genotype is what? One gene from each parent. So one allele from each parent. So here you can see both are capital. So obviously both are dominant. So here T will express itself capital T. So height of the plant will be tall. Now always remember we express the letters with the dominant trait alphabets. Here capital T is for the tallness. Dwarf we don't use D. We use lowercase t. Okay. So here you can see if capital T is with the small t also, that is lowercase t, it is the capital T which is going to express itself. So T is dominant in both homozygous and heterozygous condition. But only in the situation when both the small letters come together, then only the recessive trait of dwarfness is expressed. Okay, so let us understand this with the cross. Now here you can see the parent is homozygous tall. Right, you can see the parent is homozygous tall and homozygous dwarf. Here is a concept of purebred or pure line. Pure line plants are those plants which are continuously crossed with themselves so that they lose the recessive trait or the dominant trait completely. So they are completely homozygous for their alleles, or you can say they have the both the capital genes or both the recessive alleles together. Okay, so they are purebred. So purebred parents are taken, which are homozygous tall and homozygous dwarf. When they were crossed, gametes are formed. Now this is both are homozygous, the same kind of alleles. Here also there are both homozygous, the same kind of alleles. So this will produce same kind of gamete, that is capital T, and this will produce the same kind of gamete, that is small t. So upon the cross, you get a plant which is capital T and small t. This is the F1 generation that you have got. And this F1 generation, though it has two different types of alleles, the one which is dominant will express itself. Here is the explanation of law of dominance. So you can see that the phenotype of the plant is tall and the genotype is heterozygous, capital T and small t. Is that clear? So here you can find out that though the plant, only one variety will be seen in the F1 generation. So here ratio will not be asked. Because here ratio kya hoga? There is no ratio because there is only one variety which is formed, that is tall plant. Now when this F1 is selfed, that is when there is selfing of F1 is done, that time you will find that the F1, two F1 varieties are taken. And there you can see there are two alleles. So there are two types of gametes which are formed, right? And here also two types of gametes, similar gametes will be formed. So when they are crossed, this is the Punnett square. This is how the Punnett square is formed, where one box will give you the alleles of one parent, and here you have the gametes of the other parent. Then you write the combination, combine and write. Always remember, capital letter has to be written first, small letter has to be written later. So capital T is small t, though here is small t, but the small t will be written after capital T because capital T gets the first place to be written. Okay. Now here you can see that how uniformly the traits or the alleles have distributed themselves in the gametes without influencing the other one. So here the small t, though it was not expressive in the first generation, it will be segregated into the gametes without influencing the capital T. So this explains law of segregation. So reappearance of the recessive trait in the F2 generation explains the law of segregation. Understood? Now here you can see that there are four types of children that are formed. You can say the F2 generation that is formed. In this case, three are tall. But in what condition? The first one is homozygous tall and the other two is heterozygous tall. And the last one is dwarf. So this gives a phenotypic. Phenotypic is what? Physical appearance. So physical appearance mein you have 3:1 ratio formed, and genotypic ratio is 1:2:1, where 1 is homozygous tall, 2 is heterozygous tall, and 1 is dwarf. Now instead of writing homozygous dwarf, you can write only dwarf because recessive trait can be expressed only in homozygous condition. So you can add homozygous here, there is no issue. This is how you're going to write the answer. If a ratio is asked, you have to write the number in ratio form and then explain it in the bracket that what is 3 and what is 1. Similarly, when a genotypic ratio is asked, you have to write 1:2:1 and then again explain in a bracket that what is 1, 2 and what is 1. So this is the F1 and F2 of a monohybrid cross. I hope you understood. Here, which explanation for the F2? Law of segregation. So the two members of the pair of factors, that is pair of factors is what? Alleles or the traits. separate during the formation of gametes. So both the two factors, that is capital T and small t, will separate themselves in the gamete without influencing each other. This is also called the law of purity of gametes. They do not blend, that is capital T and small t will not blend or mix with each other. They will move into the gametes independent of each other. So this is what is called law of segregation. That is the gamete combined together in random formation. So whichever allele has gone into the gamete, then it will be decided okay, which one is dominant, which one is recessive, what should be the expression. That is the second part. First part, mein alleles going into the gamete is totally independent of each other. They segregate themselves without influencing each other. This is the law of segregation. So when a pure tall plant is crossed with the purebred dwarf plant, in F1 generation, heterozygous plants are produced. The dwarf allele, that is small t, does not blend but separate themselves, and recessive trait reappeared in the F2 generation. This explains the law of segregation. Moving to the next, that is dihybrid cross. Remember the traits, children. Here we are talking about two characters. What are the two characters that I have taken in the example? Is yellow and green. Yellow and wrinkled. Sorry, not green. Yellow and wrinkled. You can say here, look here. Yellow is a dominant trait. I have taken this, and it's a round seed. So yellow and round is the trait taken. Okay. So yellow is a dominant trait and round is also a dominant trait. So this is the seed color ka trait I have taken. Okay. And what is the recessive trait for this? Yellow ke liye it is green and round ke liye it is wrinkled. So you look at the picture, it will be easy for you to understand. This dihybrid cross explains the law of independent assortment. Here, independent assortment, we are talking about two characters. Both the characters do not influence each other. They allow each other to follow the other laws without, without disturbing or influencing each other. In the law of segregation, we are talking about two alleles. Here, we are talking about two different characters or two different genes. So in that case, the alleles were going into the gametes without influencing each other. Here, the alleles of the different characters go into the gamete without influencing each other. So that is the law of independent assortment, very important. So when there are two pairs of characters, round and yellow, the distribution of the allele of one character into the gamete is independent of the distribution of allele of the other character of the other character. Understood? Matlab yellow will not influence the inheritance pattern of green or round, not green, round. Okay, so yellow will not influence uh, the round wala gene or allele to move into the gametes. They will be totally independent of each other. This is called independent assortment. Here we are talking about dihybrid cross. You can see the example here. Your parent is homozygous yellow and round. Okay, yellow is a dominant trait. Go in party wala Y, and R is also a dominant trait. Party, these the party wala words. And here you have they are recessive trait, so green and wrinkled. Okay. Now here gametes, since they are having same type of alleles, so you have only one type of allele formed, though they will also form different gametes, but all will be same kind. So again, when they combine, they form a heterozygous F1 generation, which is same. All the F1 progeny will be same. They will be showing the parental dominant trait, that is yellow and round. Okay. Here you can see the phenotype of the F1 generation is yellow and round, and the genotype will be heterozygous yellow and round. Matlab heterozygous, this is the genotype, the genetic content. So you have to mention the alphabets when the genotype is asked, and also mention the word whether this is homozygous or heterozygous. So heterozygous, homozygous becomes the answer for the genotype when it is asked. But for the phenotype, you have to write the physical appearance. So if yellow and round is the right answer. Now you can see here I have shown you the cross between the two F1s, which will give you the different types of alleles. Now, alleles, gametes. So here, since there is two different characters being studied, there are totally four possible combinations Okay, 4 possible combinations means 4 combinations into 4, 16 offsprings are possible. So first, let us find out the Punnett square for the gametes. Now, this is also an important question for the exam. Draw a Punnett square to show the gamete formation of the F1 generation. In this case, you will put the two characters in the different box. Here you have talking about the seed color, that is seed color, and here you are talking about the seed shape. So here seed color mein you have yellow and green, and in seed shape you have round and wrinkled. The dominant trait has its alphabet. So yellow ka Y and round ka R is taken into consideration. Small y is for green color and small r is for wrinkled seed. Is that clear? So this is the Punnett square for the gamete formation. I hope it's clear. Any doubt, put it in the comment section. Next one is the Punnett square for the F2 generation. Now, since you have got the gametes, all the gametes are aligned in the same way in the one box and in the same order in the vertical place. So you have the gametes from the heterozygous parent. You can write here as this is from the male part and this is from the female part. You can also add. Then you write the possible combinations. Now children, it will be easy for you if you could have any color mark for these kind of boxes so that counting becomes easy for it. Always remember the total of whatever you get, the ratio should be 16, and 9:3:3:1 is the ratio of the F2 generation phenotypic ratio. Now here when you do the combinations for a simple calculation, you see that this diagonal will have the similar type of combinations, that is capital Y, capital R, small r, that is a parental, that is F1 ka genotype. Then this one is purely dominant and this one is purely recessive. Then you have these two as same, and there is a green color one in between. So this gives you a genotypic uh, differences. Generally, if you see the genotype, there are 9 varieties that are produced on the basis of genotype. Okay, so you can see here 1:2:1 is to 2:4:2, then again 1:2:1. Now, how will you remember this complex genotypic ratio? 1:2:1 is the genotypic ratio of the F1 generation. 1:2:1, then double it and write it in the center. Total should be 16. Similarly, in the phenotypic ratio, 3:1 is the F1 generation ka ratio. You just have to 3 times it. You get 9:3. The 9:3:3:1 is the phenotypic ratio of the F2 generation of the that is phenotypic ratio of the F2 generation of a dihybrid cross. Is that clear? Do not forget to mention what are these. Now, without doing the cross also, you can write. Now, see, the F1 generation was yellow and round, correct? And the recessive trait will be wrinkled and green. Now, the center ones will be the combinations. So, yellow and wrinkled and green and round. These are the two possible combinations. So, 9:3:3:1 will give you the possible traits where these are all the parental traits, not F1, but the parental traits. Okay, and these two are the combination of the parental traits. So, this is very important to be written in the ratio. Is that clear? Genotype of the dihybrid is generally not asked in the exam because it's too complex, it is said. But I think it's not that complex that you cannot remember. Any doubt, put it in the comment section. This is the F2 generation and the F1 generation, monohybrid and dihybrid cross. Chalo, let us see an example what kind of question it is asked. Now, a question is a pea plant which is homozygous for the green pods, which are inflated. Now, green pod and inflated pod, this is the pod character, go in, party wala, go in is here, is crossed with homozygous plant for yellow pod and which are constricted. Answer the following questions based on this. Now, give the phenotype and the genotype of the F1 generation. Without doing the cross also, if you know, if the dominant trait is green and inflated, the F1 will be green and inflated pods. And genotype will be heterozygous. So this is the heterozygous condition of green and inflated pod. Is that clear? Next question is, draw a Punnett square to show the possible combinations of the gametes that can be obtained in the F1 generation. Now here gametes so there are two traits that we are talking about. One is the color of the pod, that is pod color, and one is the shape of the pod, that is pod shape, right? So pod color is green and yellow, and the pod shape is inflated, that is fully swollen, or constricted. So you have four possibilities of gametes that is formed by the F1 generation. Now next question is, draw a Punnett square. Here they have given you to draw the Punnett square to obtain the F2 progeny by selfing the F1. So F1 gametes you have already got. Put it into the box of 4 into 4, that is 16 boxes. You can have one more layer added to show the gametes, like what I have done here. So make the possible combinations. Always remember, whenever there are two G's, they will be written together, two I will be written together. If there are capital letters and small letters, then capital letter will be written first and the small letter will be written later. The diagonals will show the similar type of F1 trait. There will be one purely homozygous dominant and there will be one purely homozygous recessive. The rest will all be the combinations. So what is the phenotypic ratio? 9 is to 3 is to 3 is to 1, where 9 is the green inflated and the last one is yellow and constricted. In between, there is a combination. So green and inflated, yellow and constricted. So it will be green and constricted, yellow and inflated. That is how you're going to write the ratios. And finally, you have the genotypic ratio, that is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So this gives you the genotypic ratio. Now this gives you the complete explanation of the monohybrid and dihybrid cross. So I hope you understood between the difference between the monohybrid cross and the dihybrid cross. Here you talk about only one trait. Here you talk about only one trait. Here we are talking about the color of the flower. You can use purple P or you can use blue B, whatever is given in the question. Here we are only talking about the color of the flower. But in the dihybrid cross, you are talking about the shape of the pod and the shape of the seed and the color of the seed. Right? Two more important questions that is asked from this chapter is, who is the father of genetics? That is Gregor Johann Mendel. And what is the scientific name of the plant used by him? That is a pea plant that we are talking about is Pisum sativum. Learn to write the scientific name. P capital, S small, and both the word has to be underlined. Okay, so this is all from this part of the lesson, as I think I have answered the question or I have explained what you have not understood in this video. For any doubt, put the comment in the comment section which video you want for the explanation. I'll be surely making it and putting it there. Right? Happy learning, children. It's already high time now. Your boards are starting soon. Have proper rest, proper sleep, and proper study. Eat well, stay hydrated. All the best, study well, and happy learning, children. Be connected with Biowise with Aanchal.